which might have kept a few people out of the meeting. These are fungi that have their spores are formed on the, what you can see in the background here, on these spine-like structures. They're not, they're shaped like spines, but they're soft. Anyways, here we go. Maybe. That's what we didn't test, is going to the next page. Oh, all I have to do is click. Okay, so what we're going to do, what I would like to achieve here is to give you some appreciation of the rather wide diversity of tooth fungi that occur in the Pacific Northwest, North America, and to be able to identify some of the common species, particularly the ones that are more notable in their appearance for one reason or another. But we'll also talk about some of the not so common species as well. So the first question that arises, well, why do some fungi produce spines? The answer is pretty much the same as for other types of structures, whether it be gills or plates or pores. The idea is to increase the spore-bearing surface as much as possible to produce as many spores as you can. And the more spores you produce, the more reproductively successful you're likely to be. So there really aren't an awful lot of ways of doing it. One is to grow plates and then grow spores on the sides of the plates. The other is to grow pores and grow all the spines, all the spores on the inside of those pores, or an inverted pore, as it were, is a spine. Now, once upon a time, there was a genus called Hydnum where all of the tooth fungi lived. Now, Linnaeus, when he published his opus in 1758, I think it was, divided the fungi into 4 groups. Agaricus, which were the gilled fungi, Hydnum, which were the toothed fungi, Boletus, which were all the pored fungi, and there was one other, but I can't actually remember what it is at the moment. Now, right from the get-go here, we get into controversy. The question is, how do you say this word? Is it Hydnum, or is it Hydnum, or is it Hydnum, or is it Hednum? Well, I don't really care too much how you pronounce it. I have consulted many authoritative sources in this matter, and essentially at the end they all sort of throw up their hands and say, well, there really is no consensus. Nothing can be claimed to be absolutely correct. We get inconsistencies in English. You get hypothesis and hypnosis, the different pronunciations, although the Y in Latin was introduced from Greek to represent a letter which, the letter upsilon, which allegedly was pronounced like the French U, as in du. So in theory, it probably should be Hydnum, but I never use that. And I sometimes, in the same talk, probably this evening, I may switch back and forth between Hydnum and Hydnum. There is a lot of confusion in the literature, even getting up to the 1980s. It seems to have settled down to a certain extent since the 90s, but a lot of references that you may look at, and in particular, one of the key references for mushrooms of the Pacific Northwest, which is David Aurora's Mushrooms Demystified, refers to Hydnum, which is now, all of those have become Sarcodon, and he also refers to Dentinums, the genus Dentinum, which has now become Hydnum. So just, if you're referring to older literature, be prepared to have to sort out from the get-go which convention is being used. Oh, right. I said originally all the Hydnums were lumped together, but they've now been split apart because they've recognized that phylogenetically tooth structures for reproduction have evolved in many, many different groups. All of the tooth fungi are basidiomycetes. There's 2 sort of main groups of macroscopic fungi, the ascomycetes, which produce their spores in sac-like structures, and the basidiomycetes that produce their spores at the ends of club-like structures called a basidium. And they're all within the basidiomycetes. All the tooth fungi are within the class Agaricomycetes, which really only excludes the jelly fungi. So it's most of the basidiomycetes anyways. But then within that, you can see that there's a lot of different orders that the tooth fungi are spread across. These are the ones that we're talking about. You don't really need to pay a lot of attention to the names of the orders and the families and whatnot. I just wanted to show you that they are spread around quite a bit. We're going to start with Mucronella up at the top here. And then the number in parentheses is the number of species in that particular genus, which is thought to occur in the Pacific Northwest. So we're going to deal first with this group at the top, up down to about here, which are kind of minimalist in terms of their overall structure, not terribly interesting to look at. Then we get some things that are a little bit more interesting. And then finally we get down to the bottom, which is where the vast majority of the toothy fungi live, the Bankera down to Hydenellum at the bottom here. Oh, and by the way, feel free to stop me if you have a question. And good luck with the technology on doing that.
When he says to contribute questions, that would be in the chat box.
We do it that way. Will I see it?
If you click on the chat control, it should pop up a screen that will show everything.
Yes, I had it open a moment ago. I don't know where it's gone. Screen sharing, participant security, stop video.
Well, if I see some urgent questions, I will bring them to your attention.
Okay, thank you. You know, somebody asked me if I was nervous about making a presentation, and I thought, no. Now I'm trying to go back one slide.
Click on
that
left.
Worked. Okay, that worked. Got it. This is the first time I've ever done a Zoom presentation, so bear with me a little bit here. The very first tooth fungi that we're going to look at here consist simply of teeth. There's nothing else to them. No branching, no substrate, nothing. They just emerge as little toothy structures directly out of the wood. These ones are particularly small. I'm just going to whiz through them here. These are common. Oh, by the way, in terms of these photos, up on the left is the scientific name, international name. On the right is an English common name, one of. There are often many English common names for things that are common, and for many others which aren't common, there is no common name at all. And in the lower right corner, if this photograph is not one of mine, it's attributed to whoever took the photograph. This is an interesting little fellow. It is really tiny. If you look at these sort of striations that curve slightly, those are the rings of a Doug fir tree. So these little fungi are about the length of them is about the width of a tree ring, an annual ring on a Douglas fir. This particular fungus was in Lighthouse Park. It was there every time I went for about 6 years, and then finally disappeared. But very hard to, most people wouldn't even notice it, but I pick up on strange colorings of things, and there it was. This is quite a common tooth fungus. This occurs just spread over the surface, unlike many of the others, which have a more elaborate 3D type of shape. We're now into a set that just has basically a smooth layer over the substrate, and then the teeth emerge from that layer. Another one of the same. I have never found this particular one, but it allegedly occurs here. getting a little beefier. And now we're into the stuff that's a little bit more interesting. Okay, Hericiums. There's been much confusion about how these are categorized of late. I've given a common name of bear's head. I've read certain references that say, oh, the bear's head refers to Hericium Americanum. There are only 4 species in North America, 3 of which occur in British Columbia. Hericium Americanum appears not to occur in BC. Okay, these things are lovely. This thing was probably about a foot from bottom to top, and they are quite delicious. They are actually in the Hericium, in the Rustiolales. Oh, sorry, I was thinking about the Hydnum, so I was going to say they're related to chanterelles, but that's another group just a little further down. Another one, the comb tooth, which is a better name for it. This also used to be called Hericium ramosum. You can see that with this one, the little spines drop down from the branches, whereas with the previous one, they sort of start on the top and hang over. And then the third species that we have in the Pacific Northwest, the lion's mane or Hericium erinaceus. I have read that this has never been found in Canada, although it's readily available in northern Washington. I personally have never seen it, but I almost never go looking for mushrooms in Washington state. Now, Americanum actually is somewhat intermediate in appearance between this guy and this guy. It has intermediate length spines, but as I say, it doesn't occur here. It's not known to occur in Western North America. Okay, most tooth fungi are somewhat soft. Some of them are a bit corky in texture, but this one is a true conch. It's really woody. It was with considerable effort that I made the gash in that one there. I'm pretty sure actually this is my photo, not, I think it may be misattributed, but I know I have tried to cut into these things and they're very hard. The toothy surface is a little bit hard to see here, but it really is. It's just like wood. This fungus, the common name you see, Indian paint fungus, was used to produce a dye, which was painted on the faces of the native people. I'm not exactly sure whether that was decorative for celebratory purposes or whether it was for combat. These little guys are always a delight to find on the forest floor. It's sort of a jelly-like fungus, but not technically a jelly fungus. You can see the cone, the Douglas fir cone over on the right side of the image here, which gives you a sense of just how small these things can be. But they kind of glow in a ghostly way on the forest floor. The common name Pacific Spirit Gummy Bear, I think, was created by Andy McKinnon. And they roughly have the texture of a gummy bear. Some people eat them, but I've tried them and they're quite tasteless. I'm not quite sure why one would do that. But they do get a little bit bigger. This one was probably a couple of inches, maybe 3 inches across, and a lot more toothy. You can see the previous one, if I can get back there, yes, has very, very fine teeth, which you can see best just highlighted a little bit on the edge, but they can get quite coarse. This one, again, was found in Lighthouse Park. I was aware of this particular tooth fungus for many years before I actually finally found one. Auroscarpium vulgara, the ear pick fungus. These days, very few people would actually know what an ear pick was. But it was a device which was used to remove wax from people's ears, and it had a long handle and then sort of a scoop on the end. So it looked very much like the fungus, or the fungus looked very much like the ear pick, so that's where its common name comes from. As you can see from the mosses that are all around, these things are actually quite tiny. They tend to grow on Douglas fir cones. Okay. Now we get into a group. This is the remainder of the fungi which are in the genus Hydnum. And until quite recently, they were all considered the ones that looked like this, Hydnum repandum. They're a good edible mushroom. The latest information based on the molecular stuff is that Hydnum repandum does not occur in North America. And on the west coast here, you get, it's replaced by a number of species, Olympicum, Washingtonianum, Meliopallidum, which can only be distinguished microscopically. So we have to apply the rather, just the generic name, Hydnum repandum group to these guys. As far as we know, they're all edible and nobody has any negative reactions to them. By the way, when I say that something is edible, it's edible by humans, by most humans. But there are things just as people cannot eat, some people can't eat peaches or strawberries or peanuts. They can have a very severe or sometimes fatal reaction. That is true of some fungi for some people. Another closely related fungus, again, we used to call this Hydnum Imbilicatum. Umbilicate because if you look very carefully, you can see that there's a hole right in the center of the cap. And in fact, that hole descends down into the base. So it's sort of like a belly button. Now, as it turns out, Hydnum Imbilicatum apparently doesn't occur, at least in Western North America either. And there are a number of names that have been proposed, but I thought I had included them here, but apparently not. Anyway, so we can still call them that. You cannot distinguish them without microscopic work and or DNA testing. I was out in the Walbrand Valley, gosh, was it 2 years ago? And we found this little fellow. You can see that unlike the previous one, which has a bit of a scurfy cap, this one is actually very smooth on top, and there was no evidence of an umbilicus at all. So we photographed it carefully, collected a sample of it, and I gave it to somebody who was going to be doing DNA analysis on the ones that, the specimens that she brought back. And I actually just asked a few days ago, so whatever happened about that thing? And she said, oh, no, I never did a DNA analysis on that. So we don't know which of the many possibilities it is. Okay, last in this general group is Cystotrema confluens. I've never found this. It's a pretty good size, as you can tell by the surrounding vegetation. No common name because it's not at all common. Okay, we're now, so we've sort of quickly run through these first groups here, and we're now into the last group, which is in the order Thelophoralis, which contains only two families. One is Thelophoraceae, which are little blade-like fungi that grow on the ground, and the Bankeraceae, which are all toothy fungi. So here's a few of this is where most of the, I think, really interesting fungi are amongst the toothy ones. First off, it's Ectomycorrhizal. And for those of you who may not be familiar with that term, the myco is fungus and rhizal is roots. They form a symbiotic, these are fungi whose hyphae, the little parts of the fungus that are underground, form a symbiotic relationship with, can be herbs, bushes, and trees by wrapping their hyphae around the roots and exchanging nutrients from the soil, which they are actually much better at extracting. The fungi are much better at extracting than the plants. But in turn, because fungi can't produce their own food, they have no chlorophyll, so they get sugars from the tree. So there's this little economic trade that goes on between the fungi in the ground and the plants. And in fact, plants that do have this ectomycorrhizal association with fungi grow much, much better than ones that don't. I mean, even the same species, if you sterilize soil so that it's full of nutrients but not fungi, those plants that are planted in that soil will not grow nearly as well as ones that are planted in soil that has these ectomycorrhizal fungi. The Bancheriaceae, the tooth fungi in this group, are mostly associated with conifers, particularly out here. Back in the east and in England, they're much more common. Some of them are more commonly associated with hardwoods, but we don't get some of those species out here. They have spiny, warty, enameloid spores. Enameloid means they don't turn bluish with a particular chemical, Meltzer's agent, or iodine. And unfortunately, well, only one of the species is considered edible, but it's quite rare, so you really shouldn't eat it. It's probably going to be on the red list if they can ever get that sorted. Now, here's a statement that was made as of 1986 by Baird. I forget his first name, Richard, I think, Richard Baird. He's out of the University of Florida. And he has spent much of his career looking at these toothy fungi. So there is still believed to be confusion as to the correct species names used for North American taxa. Mas Easteranus, who was a world authority on toothy fungi, discussed many North American tropical and European type specimens. These are the ones that are representative of what any given species is supposed to be. And he concluded that many of the North American species were incorrectly named, and thus may be synonyms or new species. Well, that, I suppose, would be considered foreshadowing. It certainly is in this case. The first thing you want to do if you're trying to identify tooth fungi is make sure that it really does have teeth. I have had numerous people bring me a fungus and say, what is this tooth fungus? And sometimes I've gotten confused. If you look at it carefully, in fact, although it maybe looks like it's got teeth, if you look at it very carefully, often it's a polypore. So this is Onia tomentosa. This looks actually quite like one of the ones that one of the tooth fungi that's coming up that we're going to look at. Its texture is very similar. Its color patterning is very similar. Surface texture is a little different. Another one that's very commonly mistaken for a tooth fungus is Fiola Schweinzii, the Dyer's polypore. It grows very commonly. It's a rotter of Douglas fir trees. It causes a lot of timber loss. So it's very common, so people find it a lot, and they think it's particularly if they're looking for toothy fungi, they think that's it. In terms of warty versus spiny spores, these are some electron microscope images. The one in the upper left corner is basically what they mean by spiny. The rest of these pretty much are what you would consider warty. It turns out that the spines and the warts are really not very helpful in distinguishing even the genera sometimes. So I put it there because that is one of the characteristics they use, but not that helpful. And things have changed. Okay, now one interesting thing, by the way, is that these fungi appear to occur almost exclusively in the Northern Hemisphere. I have combed herbaria records, literature, and whatnot for members of the Bancheriaceae in South America, and I think I've found 2 reports of these things. So it could be that they're just not part of the fungal flora there, or it could be that those ones have been introduced. I don't know, but they are extraordinarily rare down in the neotropics. So here's what if you want to look at these guys, here's the things you should look at to help try and identify them. The first is the habit of its growth. So there are two categories. One is what's called determinate, and they sort of look like mushrooms, except for the fact they have teeth. And the other one is indeterminate manner of growth, and they look more like tops. Now I'll show you an image which shows you what I mean by indeterminate as the next slide. The flesh texture is either soft, which often goes with the determinate shape, or corky, which goes with the indeterminate shape usually. The smell, some of these things have very distinctive smells. The taste, I don't know if you've heard, but you cannot have any ill effects, that no one knows of any ill effects that you can have from touching fungi, handling them quite extensively, and you can actually take a bite of one and chew it up, and as long as you spit it out, you can't possibly ingest enough, even if it is a toxic mushroom, that it will affect you. So tasting mushrooms is a very common way of helping to identify them. And the tastes, the notable tastes for this group is they're mild, or they're acrid, or they're bitter. Those are the three primary tastes that you'll encounter. Obviously, the color, we respond, human visual system and brain responds very much to color, particularly of the cap, of the spines, the teeth, including the tips, which are often a different color, the color of the stem, the color of the spores in theory, although it is really hard to get a spore print from these. Spore print is where you take a mushroom, typically if it has a cap, you take the cap, cut off the spine, lay it on a piece of paper, white or black, or actually I find it best to lay it on top of something that's half black and half white. And then you leave it for several hours and you come back and the spores will drop onto the paper and typically will leave some sort of a color. But then something that's not so obvious if you found something and are looking at it, maybe picked up and are looking at it, is what color is the flesh? It can be quite key to cut them in half vertically through the stem, and the coloring and patterning internally can often say a great deal about it. The last field character that's very important is the nearby trees, and as we go through the various species, will indicate that certain fungi are associated, certain of the fungi are associated with certain trees and others with other trees. Now, determinate versus indeterminate. Determinate growth is the fungus basically has all its cells and it simply absorbs water and swells. And as it does so, if it runs into anything, it simply pushes it out of the way if it can, or if it's sort of immobile, then it will sort of bend its shape. It will become somewhat distorted, but it can't. There's nothing else it can do. All right, it just swells. It gets bigger. Indeterminate form of growth is where it keeps adding cells, and so anytime it encounters something, leaves, sticks, stones, whatever, it simply adds more tissue and eventually will engulf whatever it's encountered. So here you can see that these sticks are actually now protruding from the from inside the flesh of the fungus. So because it engulfs whatever it runs into, if you find any of these, don't stand too close for too long. All right. I'm going to talk about the way you will find the members of this family divided into genera conventionally, because this is what you're going to find in pretty much every book that you see out there. And then we'll tell you how things have changed. First, the genus Bankera, there's only two species known. They look like mushrooms. They're soft. They have determinate growth. They look like a mushroom. they have white spores. Phelodon have the other form. They're turbinate. They look like a little top. They are indeterminate. They will grow around something, and frequently the individual mushrooms will grow into and fuse with each other. They have kind of a corky texture. They you can cut them with a knife, but you have to do a little bit of work to get through it. And both of those two, you notice they share all the three characters here. They smell like fenugreek, which is like soup bouillon cubes. And ordinarily when I make this presentation, it's live. And so, you know, here they are. You take a sniff and see what you like. I mean, I want to go and have some soup as soon as I smell this. And the drier they get, the stronger they smell. And the smell lasts for years. So I've had some of these things that I've had for years. And you, in a container, open up the container, and the smell just wafts out. Unfortunately, they're not edible at all. They smell good. All right. These other two, Sarcodon and Hydenellum, are brown spored. They don't often have, well, there's a couple that have an interesting smell, but by and large, they don't have much by way of smell. There are some interesting tastes that we'll talk about. Like the Bankera and the Phelodons, you get the white spored ones. There's a soft group, which are the Bankera. The corky ones are the Phelodons. And within the brown spored group, the soft ones are the Sarcodons, and the corky ones are the Hydenellums. Also, the Sarcodons are mushroom-shaped, and they're determinate in their growth, and Hydenellums are top-shaped and indeterminate in their growth. What we looked at previously, this is a Hydenellum. Okay. So this is one of the very few ones that's edible. Bankera fuligineo alba, the drab tooth fungus. It tends to grow in association with, what do I hear you say? Yes, pine trees. It has a character that it's kind of a sticky surface, and the junk tends to adhere to the surface. So that's one of the ways of distinguishing it, because this guy and the next guy, oops, same, it's just an internal shot, back it up. This fellow can be often distinguished just by noting that on this guy, which grows in association with spruce, it doesn't tend to accrete anything on the cap. So, and with these, always a good idea to cut them in half to look at the interior texture. The interior texture is not very interesting in this case, either for Violescence. Both of these are very rare. I did find one specimen in about 20 years. I found one specimen of Bankera violescens up in the place where they built the ski hill south of Whistler for the Olympics. I can't remember the name of the valley offhand. And I've...
Callahan Valley.
Yes, that's it, Callahan. Thank you. I found a couple of these, but people tend to bring them into like the Vancouver Mushroom Show. Every once in a while at a mushroom show, people will bring them in. Okay, the Phelodons, the other white-spored ones. Notice in this case, more top-shaped, and you can already see the determinate growth here. And here, the individual fruit bodies have grown into each other and coalesced. So they've kind of engulfed. It's a mutual engulfment. This is the particular specimen that I found just about 2 weeks ago up here on one of my first walks in the forest here. This is the one I was showing you. In fact, this is one of the specimens that I collected on that particular trip. The rest of them are all packed away in boxes somewhere or other here. Yeah, for those of you who don't know, I just moved, so I'm not quite sure where a lot of my mushroom resources are. Okay, the other species, well, sorry, there's only 3 species in this area, as far as we know. The other one is Phelodon melaleucus, mela being black and leucus being white. And it tends to have a bright white margin and a much, much darker center than these guys. These tend to be quite a lightish brown and quite distinctive zoning. These ones, less so. Finally, there is Phelodon atratus, which I have never found. It's sort of a bluey purple, very dark bluey purple color. Yes. All right, that's it for Phelodons. Only 3 species. 2 Bankeras, 3 Phelodons. The vast majority are of the toothy fungi in the Bankeraceae are in the genera Sarcodon and Hydenellum. Now, if we could flip back to this slide, we see that there are maybe 16 species of Sarcodon that have been identified here, and 13 species of Hydenellum. It's very hard to identify a lot of these things. Some of them are dead easy, like this one. Many others, they almost seem to integrate, and there, in fact, is suspicion that, well, first off, with the indeterminate growth form of the Hydenellums, the shape can be pretty much anything, and coloring can be quite variable, and they do seem to almost intergrade. So it can be really hard to match a specimen to a description, and there's been a lot of confusion amongst the people who study these groups over the decades. Anyways, Sarcodon imbricatus is the type specimen for the genus Sarcodon. It's very distinctive. It's quite large. It can grow up to be almost about 8 inches, 20 centimeters across. It has these really noticeable scales, upright scales, large scales, particularly in the center of the cap. They tend to be upright. It tends to dip in the center, become somewhat depressed in the center. This particular species in Europe, in a study that was done in Europe, is associated with spruce trees. There's another species that originally, for the last 50 years or so, people had actually lumped 2 species together. Although earlier mycologists had recognized a difference, somehow or other they got blended together. And the other one has been separated out, and it now sort of more carefully distinguished. It goes by the name of Sarcodon squamosus, and it is associated with pine trees, at least in Scandinavia, which is where the study was done. Now, that has led a lot of people to look at a photograph, and if they see pine needles in the photograph, they will say, oh, that's Sarcodon squamosus. The problem with this particular setting is that most of the trees where this photograph was taken up in Manning Park is a heavy mixture of lodgepole pine and Engelmann spruce. So you don't know what it's growing in association with, regardless of what the needle cover is like. Anyways, pretty sure from the characteristics, particularly the depressed cap in the center, that this is, in fact, imbricatus. I'm not going to show you squamosus because I have yet to find any reliable report of squamosus in North America. It may occur here, but we don't know. Okay, the hawkwing, that's how it's known. Another difference between imbricatus and squamosus is supposedly the taste. One being somewhat bitter and the other being quite mild and good to eat. However, I have sampled one of the first ones of these that I found. I sampled it and I found it to be quite bitter, but then I had somebody cook some up and it actually tasted quite good. But this was way before anybody had published this article about the difference between imbricatus and squamosus. So maybe they had found squamosus. I don't know. There are mysteries that remain. This is another very common one and quite distinctive, for a couple of reasons. The first is that you get this blue-green at the base, and also it tends to grow in sand, so you get a lot of the sand adheres to all these little rhizoids at the base. So Sarcodon scabrosis, the bitter spiny cap fungus, and I can attest to that. That's a good way to test it. Take a little test of a little bit of it, chew it up, you'll get that very bitter taste, and then you just spit it out, and you're fine, except for the fact that often these bitter tastes last for 5-10 minutes or so. And lastly, this I don't find very often, but always a pleasure to see on the forest floor, the violet hedgehog, Sarcodon Fusco Indicus. Now these are the, this particular one, by the way, is very interesting because if you look very carefully, I don't know how clearly you can see some of these images, but a lot of the spines on this one are forked right at the end. You see that just as the spine is brown, and then just as it changes color to be light, it forks into two subspines. I have seen this on a couple of other tooth fungi, but it seems to be a rare condition. No idea what produces it.
Yes, I can see the fork.
Oh, good. Okay, now have a good look at this and see if, to your mind, this differs considerably in terms of its growth form or mushroom-like habit from these guys. We'll come back to that. I'm only going to cover these 3 Sarcodons, but then we're going to revisit them as we get down to the end. Oh, sorry, a fourth one, but we don't know what this is. Every year, if you go up right now to Manning Park and down Memaloose Trail, about 1.5 to 2 kilometers, there's a spot which is chock-a-block with these fungi. They're quite large, as you can see from the quarter that's sitting on top of the cap. It doesn't seem to match any description that I've seen. It's somewhat close to Sarcodon livigatus, it's close to Martio flavus, but it really doesn't match that well. The spines are super long. Many of the spines on these fungi are 3-4 millimeters. Almost all of these, or at least, well, you get right out to the edge, they're shorter, but close to the stem here or in the center of the cap, they were almost all over 10 millimeters, and some of them as many as 15. So very, very long spines. All right, now we're going to move into Hydenellum. Now, many of you have probably seen this. Among the tooth fungi, this is the Amanita Muscaria, the fly agaric of the tooth fungi. Like, if somebody asks for an iconic image of a mushroom, 80% of the time it will be Amanita Muscaria with the red cap and the little white warts all over it. Well, I don't know, people I guess respond to red and white because this, among the tooth fungi, people tend to know this. Strawberries and cream, good description based on the color. It is also known, however, as the bleeding tooth, and one can understand that as well, but it's a bad name for reasons that I'll explain momentarily. Another look at it, this is very young. It's just emerging, and it sort of comes out as a big, almost like a, a bunch of uncooked bread dough, starting to develop on the teeth. Here it is starting to attain more of its character, again, the turbinate shape. These were wet, it had been raining a little bit, so all the droplets sort of had spread out. And here it is a little bit older. And you can see that it's lost the reddish tint, but you can still see in the bottom right corner here a little bit of the scrobiculate nature, the guttation that's produced by those drops. And oops, this is what this thing can look like when it gets older. Here is the interior texture of one that's fairly young, so you still see a lot of the red fluid in the top, but internally it is just a dull, darkish brown. Now, oops, okay. When it gets to be this stage or later, it tends to look very much like this. And most people don't realize that this is one of these strawberries and cream fungi. This particular one is not. I'm simply saying that when it gets older, it tends to look like this. So how do you tell without the drops if it's the Hydenellum pecii or whether it's Hydenellum scrobiculatum? Well, it turns out there's a very easy test, which is to chew a little bit of it. A very little bit of it. In fact, all you need to do is press your teeth typically into the edge of it if it's fresh, and you will, about 5 or 10 seconds later, there's a little bit of a delay, you get this extremely hot, acrid taste, which lasts a good long while. It's a wonderful experience the first time. Okay. You remember I mentioned that it's important to look at the flesh for these things? That's another key. There are some things which can look like that. You cut them open, they can look quite different on the inside, but Hydenellum pecii looks just this dull brown in the interior. As it gets older, it loses the whitish tinges up here, and the red, of course. Now, you'll notice that up at the top here, I've labeled this pecii/diabolus. There's been a controversy going back and forth as to whether there are one species or two. Pecii is allegedly associated with conifers and diabolus with hardwoods. Mascisteranus, the world expert that I mentioned earlier on, concluded that they were in fact the same species, but people in England in particular distinguish them. So I've never seen this growing anywhere around hardwoods, so I suspect that we only have the one kind here. Okay, scrobiculatum, again, very similar in appearance, but a scrobiculate surface. Now, my comment earlier about the bleeding Hydenellum being a bad name for Hydenellum pecii, this is Hydenellum aurantiacum, the orange Hydenellum. And at an early stage, it will sometimes produce orange droplets, as you see here. This one is young. This one is a fairly typical appearance of one that's older. But it's also the case here that either it's highly variable in form or we have a number of closely related species. The next one is quite different in that it has all these seemingly separated caps growing together. But if you look at the texture right down on the bottom here with all those sort of lumpy outgrowths, very similar to what you get here, this probably is in fact a Hydenellum aurantiacum as well, although I have seen things that look like this that were claimed to be another species called Hydenellum christina, I think it is. Yeah. Again, if you get one of these things, cut it in half, see what it looks like. Hydenellum aurantiacum is uniformly bright orange all the way through, although you can't really tell that from looking at the outside. Okay, so pecii with the red drops, Hydenellum with the orange drops when they have drops, but very orange. There is another orange species which does occur in the Pacific Northwest, although I've never found it. Much more smooth and sedate looking than aurantiacum. I said than Hydenellum aurantiacum, but I include it for sort of completeness here. Keep your eye open for this one. And those are ones with reddish tones. Now we go into, there's a number that have bluish tones to them. Hydenellum caeruleum. And the bluing can be quite subtle, often occurring just on the growing edge of specimens. Here's a photo taken from somewhere in Europe where the blue is much more pronounced. But when you cut them open, the base of it looks very much like Hydenellum aurantiacum, but it has these bluish tones in the upper growing areas. And when you find one of these that's questionable, Again, cut it open, and you see this pattern, you know what it is. I'm actually quite surprised at how resistant people are to cutting these things open. On Facebook last week, somebody took a photograph of something which was clearly a Hydenellum, but the coloration was very unusual, nothing I'd ever seen. And they took the usual photographs, and I put a comment in that said, please, if you've collected it, cut it in half and post a photograph of what it looks like on the inside. And the person went to the trouble of collecting it, actually going back into the field and collecting it, but then didn't cut it and post a photo. But she did keep it for me, so I now have it, and I'm going to look at it. Okay, Hydenellum suaveolens. The upper part, the cap, belies its nature because it's kind of nondescript. This is sort of brown and white. But the stem is this wonderful purpley color, mauve, purple. And some people actually use these as air fresheners in their car. Probably only mycologists, but it smells very much like an air freshener. It's just got an absolutely wonderful smell to it. Unfortunately, I didn't know about cutting them in half at the time I found this one, which was a lovely representative. But when I was out in the Walbran, we found one. It's not as purpley, but the internal texture is quite similar. It's got more like indigo, I suppose. And I've seen a few cross sections like this, and they seem to be very strongly striated like this. I don't know whether those are annual growth rings or rain versus dry. The regularity suggests to me that this might be annual growth, but I don't know. Okay, the regal Hydenellum. What can I say about this? If you go to Manning Park around this time of year, again on Memaloose Trail, you'll typically find a whole bunch of these. I don't actually know whether this was photographed in Memaloose, but it was definitely photographed up in Manning Park. Interior texture, quite interesting. Very dark in the lobes, but this sort of map-like texture with irregular regions that are, some of them very creamy, almost white, some of them are kind of orangey. A very interesting pattern, but helpful for the purposes of identification, as I will indicate in a moment. Okay. This photograph, I was trying to find something earlier today, trying to match something that looked like this. And I knew I had seen a photo somewhere of it, and I was looking for it. And I came across this photo that I had taken in Manning Park, 2007. And I thought, oh, this is Hydenellum cyanopodium, because it's got bluish teeth. And then I noticed the stem that has this cream orangey colors, and I realized that, no, that's this. So this is, in fact, Hydenellum regium. But when it's very young, it can have this very compact, blobby form to it. I had recently seen a photograph of something that looks, except for the droplets, very, very similar. This was again posted on Facebook in the South Vancouver Island mushroom identification group, and somebody else, Leah Bendlin, who's a member of the Pacific Northwest Key Council, said, well, I'll put $2 on it being Hydenellum regium. And at first I was dubious because I had forgotten about this sample that I had collected over 10 years ago. But I think indeed that probably is it, and notice that it also is producing these droplets. I actually read in the literature that Hydenellum chiralium can also produce droplets, but it produces red droplets. I wonder, in fact, if all of the species produce droplets under the right conditions, but I have no idea what the conditions are. A couple more. Hydenellum concrescens, sort of heavily zoned, but in a very nice kind of way. These tend to be quite nicely round, but with these little outgrowths at times. What it looks like from the underside. An extraordinarily rare one here, Hydenellum Geogenium. This actually is a rather pale variant of it. I've never found it. In fact, I think it's only been found 4 times ever in the Pacific Northwest. In 2 of those locations, the location no longer exists. One's now under a parking lot. I think they've built some buildings on top of the other one. Scott Redhead, who was the curator of the National Herbarium in Ottawa, found some on Haida. And there was some found somewhere else. I don't remember where else, but that's it. Very, very rare. Unfortunately, the only decent photo I could find was this, and it's not very yellow. All right. Now, the Vankereaceae taxonomy reconsidered. There have been a number of papers produced more recently. So going back to Baird, who remember in 1986, who was saying that, well, it seems that there's some problems with the identification of these things. Comparing the European names probably don't apply to North America. Maybe there's new species here. Okay, so many species within Hydenellum and Phelodon have an indeterminate type growth, often dependent on environmental conditions, making discrimination between closely related species difficult. I can attest to this, without supportive molecular sequence data, which unfortunately I don't have. Oh, that was his 2013 paper. And there, he has concluded, along with some others, that Bankera is really just synonymous with Phelodon. If you look at the DNA stuff, they intermix. And so they really have to be considered a single genera, a single genus. This is a bit problematic because on the one hand, you have the determinate growth form and the indeterminate growth form. And they are distinctly different in terms, well, at least I find them distinctly different in terms of their texture. One is very mushroom-like, just like it's sort of the texture that you'd get from this grocery store mushroom. And the other is really corky and hard to cut. Further, published just last year, although Hydenellum and Sarcodon do indeed appear to be distinct genera, if you look at the phylogenetic clades based on the molecular work, but the current division of species into these two, which is based on the texture, Basidioma texture, Basidioma is just the fruiting body, makes Sarcodon paraphyletic with respect to Hydenellum. So it's mixed up. Sarcodon has things which appear to belong with Hydenellum. Consequently, some name changes. So we looked at 3 species, apart from the one that we couldn't identify, well, I can't identify, the big one at the end. We looked at Fusco Indicus, Imbricatus, and Scabrosis. Imbricatus is the type species for Sarcodon, so it has to remain Sarcodon. But S. Fusco Indicus has now been shunted over into Hydenellum, so it becomes Hydenellum Fusco Indicum. And Sarcodon Scabrosis, again, very mushroom-like, very soft texture, is now Hydenellum Scabrosum, in with the things that have this much harder, corky texture and this indeterminate growth form. Are you confused yet? If you're not paying attention. A couple more I found, again up in Manning Park, lots of toothy fungi up in Manning Park. Wonderful thing. This is the photo that I used earlier to indicate what is the difference between determinate and indeterminate growth. This is the indeterminate growth. This appears very like Ferruginum, which is synonymized with Pineticola, whence the name, the pine-loving Hydenellum. Pineticola means pine-loving. Well, we certainly seem to have that. There certainly are some pine needles in here, although this too has a bunch of, it was growing amongst Engelmann spruce, so who knows. But in the overall appearance, I never collected the specimen. I took this very early on in my beginnings of study of these things. And so I'm just really kind of guessing from the this photo and a couple of others that I took of it. But and that's why the question mark up here at the top. But it looks very much like Hydenellum ferruginum, rather drab in terms of its coloration. And then there's this. I've been looking at this thing for years. It pops up in the Vancouver Mushroom Show about every other year. I went down to Hoypus Point Park near Deception Pass in Washington. The woods were full of it. In fact, this particular specimen was collected there. And it's just got a zillion of these little caps with the little stems which all are fused into a base, although they do tend to remain somewhat distinct even though they're kind of fused together. I know that seems contradictory, but imagine a mass of tightly bound but individual stems. Eventually, I found a photo in the back of Kenneth Harrison, who was the leading Canadian researcher in these kinds of fungi, from a publication of his in 1961, I think, the stipitate stem-bearing hydnums. At that point, they were still all hydnums of Nova Scotia. And there's a photo in there that's a dead ringer for this. It's what they call multiceps. Now, I have seen some photos out here identified as complectipes, which is a species, a new species name given by researchers out of the University of Washington. Hall and Stuntz, Dennis Hall, I think, and Daniel Stuntz. The problem with those particular species is that Hall did his PhD and studied this particular group. They produced a very nice series of articles back in 1971, 72, talking about all different kinds of tooth fungi. And then, of course, when you create a new species, it's very important that you take the particular specimen that you used as the basis for the description, and you put that in a herbarium so that if anybody else wants to compare something to that, and particularly nowadays, you can do a DNA test on the original specimen and compare it. Hall went off, and nobody seems to know where he went or what he did, but he took all his type specimens with him, which makes it very difficult to know whether complexipes and multiceps are the same thing or not. So, the realities of mycology. Okay, so that's all the ones I was going to talk about. I've been going on for the better part of an hour, so a good time to call it quits. If you're interested in doing a little bit more, Anne mentioned in her introduction the Pacific Northwest Key Council. The keys are downloadable from this site, SVIM, South Vancouver Island Mycological Society, and there are a couple of things, a couple of them that are devoted to tooth fungi. Also, I've just discovered the Puget Sound Mycological Society has an excellent pictorial guide to a lot of tooth fungi. So if you're interested in looking at these a little bit more and some other species that I didn't cover, I encourage you to go there. I don't have the link here, but if you go to PSMS and tooth fungi, you'll probably find it. It has a very strange, the top level domain is Alpentel, and I have no idea what that refers to. Anyways, there you go. I hope you keep your eyes peeled for these guys, and then we'll have some appreciation for them when you see them, when you're out in the woods next.